All right. So so far we talked about the carbohydrates and the glycoproteins, which are one of the critical macromolecules in the biochemical systems. Now we're going to move on to the different type of macromolecule, which are lipids, and those lipids forms membranes, cytoplasmic membranes, plasma membranes, as well as some membrane transport system too. Okay. So first, we're gonna talk about the definition and the properties, chemical properties of the lipids, again, which is one of really critical macromolecules. Lipids are a class of biomolecules with a hydrophobic character. So hydrophobic meaning that it's water hating. So that's basically have a poor solubility to water, but it plays a critical role in the cell structure, energy metabolism. As lipid is definitely involved in generating energies as well as the storages of those energies in the different forms, right? So the storage and the signaling too, like cell signaling pathways and so on. And lipids are major structural components of the biological membranes, which is really critical actually. So again, it's connected to this first role that is listed here as a cell structure. So, and this biological membrane basically serves as organized biological processes. So, because the many of the biochemical reactions has to happen at a certain region for the accelerating their processes, as well as you have to raise the local concentration of those molecules that are interacting to raise the possibility or the probability of that reaction that is really occurring. So those molecules have to be basically compartmentalized, and the cell membrane basically does that job. It doesn't just simply apply to the plasma membrane, but also applies to all the organelle membranes like the nucleus, ERs, Golgis, and stuff like that if you're familiar with the cell biology. And these biological membranes also keep cytoplasm separated from the organelles and extraplasmic compartment. We already talked about this, and this is a crucial requirement for maintaining, sustaining the life, basically, right? Separating from the environment. And not losing the most like precious stuff basically out to the environment. And these biological membranes are not totally impermeable. So it's not like a Ziploc bag basically that where it has some permeability depending on the molecules that you're actually looking at. So those membranes allow regulated the flow or the exchange of the molecules via gates and pumps and channels and so on. Most are consists of the proteins, and also biological membranes allow diffusion of some molecules, especially some like nonpolar molecules that can easily stuck and embedded to the membrane can go through that by the diffusion and gets inside the cell or gets outside of the cell basically. And the second function, second role that we talked about the liquid lipid is the energy metabolism and the storage here too. So energy storage function, and an example of using the lipids as the storage is the adipocytes or the fat cells that basically generates and stores the energy in the form of lipids. And also involved in this signaling pathways. So examples of those lipids, that are involved in this cell signaling pathway functions are the steroid hormones, sex hormones, or the growth hormones and stuff like that, prostaglandins and diacylglycerols. That are involved in many signaling pathways, including kinase signaling pathways. It basically cascade the signaling pathway that involve the multiple steps are involved for changing the conformational changes or the property or the function, adding or the removing the functional groups of some proteins that are involved in those pathways, and the lipids are involved there, at least for initiating that pathway, or they also work as a substrate for them. And these are the simple structures of the lipids. And they have all different classifications. And we're going to go over each classification actually, and talk about their structures, their chemical, physical properties, and so on to better understand how these lipids are used for biochemical reactions and sustaining life, basically. So again, lipids are the biological molecules that are insoluble in water, but soluble in nonpolar solvents, like oils, basically. And these are the different classifications based on their structures in the lipids. The first one's fatty acids that contains a polar carboxyl group as a head. So it's going to be COOH or COO minus. And you have a non-polar hydrocarbon chains like CH, those kind of things as tail. And the second classification is triacylglycerols, which are used as a storage form for the fatty acids. And that basically stores quite a lot of energy. And we briefly talked about the efficiency of storing energies in lipids compared to the carbohydrates. And the third one is glycerophospholipids, which are the major components of the biological membranes. And again, we're gonna delve into that a little later and talk more about the subgroups of these glycerophospholipids and how they're involved in forming the membranes. And sphingolipids are also important that contain 18, specifically 18 carbon and also contains the amino alcohol functional groups as backbone. And they are the important components for biological membranes too. Examples are sphingosine. Sphingosine ceramides are basically the derivatives. They are critical, forming the critical portion of these sphingolipids, as well as sphingomyelins of cerebrosides and gangliosides and so on. Again, we're going to actually discuss all these different molecules and their structures in this lecture. And waxes are a different type of lipids for the classification. It has a long chain of alcohol and long chain of fatty acids that are esterified. And you can actually see the structure here that has here's an ester, and it actually links to these two, carboxyl, the alcohol, and the fatty acid here. And terpenes have the isoprene as their building blocks or building units, and also important for the making some kind of chemical features, especially for the plants. And steroids, as you know, are the really interesting and characteristic fused ring structure that has like a tetracyclic structure. Actually, has the four rings, carbon-based rings that get together to form this steroid structure. An example is cholesterol, which is the most common form of steroid, and that serves as a precursor of many important biomolecules, especially hormones. And I didn't actually mention about these structures, but here you're seeing the triacylglycerol structure, steroid, like I told you, and the other functional groups like sphingolipids there. The first one on the classification that we talked about the lipid is fatty acid. And that basically, like I told you, have the carboxylic acid group, which is a COO minus, you can see right here, as well as all other fatty acids basically, and also have the long hydrocarbon side chains like that. So this carboxylic acid group forms the polar head because it's negatively charged, so it's basically will be polar, meaning that it can have affinity to the other polar molecules, including water molecule. And also it has a nonpolar tail, which comprises the hydrocarbon side chain. And this carboxylic acid group, carboxylate group, is ionized at the physiological pH. That means instead of staying as a COOH at a physiological pH, which is around 7 or so, it'll become as a COO minus. And the length of this tail, nonpolar tail, ranges from 14 to 24 carbons, but most common ones are C16 and C18s. And as you can see here, oops, Is it working? Okay. Some of them are just with the single bonds, like this, all of them. But some of them, some of the other fatty acids contain double bonds too, one or multiple. And we're talking more about the structure of this fatty acid now, especially taking into account of these double bonds and their locations, their numbers too. So here are 4 examples of the protein, the lipid structure. The first one is a stearic acid. As you can see, it has the carboxylate on the top here, and the carbons are basically numbered from this carboxylate. So this first carbon will be numbered as 1, and then 2, 3, 4, and so forth to the 18th, basically. And oleic acid is right next to it, and as you can see, it contains the same number of carbons and also carboxylate. But instead of having all the single chain, the single bonding for in the stearic acid, it contains 1 double bond between 2 carbons at C9. And here's a linoleic acid that have 2 double bonds, or the triple in the linolenic acid. So let's go back to this text here. Stearic acid or stearate that we looked at on this right here has the melting temperature of 69.6 degrees C, and it's pretty high. And melting temperature is basically where you can see the oil or this lipid changes solid to oil from the solid form to the liquid form, basically, and that's 69.6 degrees C. So basically, at room temperature, stearic acid would be in the form of what? Is it solid or liquid or gas? It's going to be solid, right? Because it has a really high melting temperature. So it has to get to the 69 or 70 degrees C to actually melt it down to oil form. So it's solid. And the shorthand notation for the stearic acid is 18, 0. And 18 means that the total number of carbons that comprises this fatty acid. And the number after this colon means that how many double bonds it has. So an example for palmitic acid is 16, 0, because it has 16 carbons, but without any double bonds. The next one is oleic acid here. The melting temperature, the TM, is much lower than stearic acid, as you can see. It's the 13.4. So at a room temperature, like 20-ish or so, is it a liquid or is it a solid? Liquid. Now it's going to be liquid. And when you look at the structure, it's pretty much the same. The only difference here is the existence of this double bond, right? But it makes a huge change, as you can see here on the melting temperature. The short end notation is 18.1, of course, because it still has 18 carbons, but also contains 1 double bond here. And that's at C9, between C9 and C10, right? So you actually use parentheses right after that and put 9 to indicate the location of that double bond. Okay? What is it? Oh, I see. Why? So when you look at the structure, there's another double bond here between C and O, right? Why is it not counted? Why not? Exactly. This does not form the hydrocarbon or the tail. And what we care is the tail, right? But this COOH is a common part that works as a head group. It's a carboxylate, right? So of course, it contains double bonds for the oxygen atom. So it's not going to be counted. I mean, it's just always there. So does that? Okay. So And let's move on to, again, just containing one double bond makes a huge change in the melting temperature, and of course, the behavior of the molecule too, right? And the third one is linoleic acid here, which contains two double bonds. So the melting temperature even go lower from this 13.4. So it's negative 9 degrees C. So of course it's gonna be, it stay as a liquid form, even at the freezing temperature for the water molecule. And the short notation would be, can anyone try? What would be the shorthand notation for linoleic acid? Exactly, 18, 2, and in parentheses, you can write 9 and 12, right? It's quite simple, but can get you a lot of information, right, without actually writing out this whole structure. And the last one is linoleic acid. Of course, it contains 3 double bonds, so the melting temperature even go lower than the what linoleic acid has, and the shorthand notation would be 18, 3, 9, 12, 15. All right? And this double bond definitely gives us an interesting feature there, interesting property there. And this double bond is basically called as unsaturated state. So whenever you see the fatty acids containing double bonds, you can call those fatty acids unsaturated, because the saturation meaning that all the valencies were saturated, or basically occupied by making the hydrocarbons, as opposed to this one still has the valency available there, right? So there's a room for that, so it's not saturated yet. And when you look at these double bonds, they're typically cis. Cis meaning that these all double bonds are on the same side of the fatty acid molecule. This is left-hand side, and 9, 12, 2, 9, 12, 15, all those things on the same side, right? So that's cis. As opposed to if you're gonna see, in the different direction, then you can call it trans. And you've heard of, trans fat basically, which is pretty bad, right? Because it's has a lot of, different properties, but it's hard to make it actually. And since it typically, has the cis form, it's gonna have this double bonding every third bond. So 9, 10, 11, and then 12, right? And then 15 and so on. So it's gonna be every like a 3 bonds or so, third bonds or so. And several double bonds can exist in the polyunsaturated fatty acids, and it can affect again the behavior of the fatty acid molecule, as well as their properties, especially for melting temperature. And there's almost never cases where there's a conjugation like alternating single and double bonds again. It'll create the trans conformation instead of cis. So it's really hard because of the steric clashes there. Any questions so far? The fatty acids are a lot actually, because as you can imagine, I mean, you can have 12 carbons in your hydrocarbon tail, but also have 24 or so. And here it lists all the saturated fatty acids and their names here. We already talked about palmitic acid, stearic acid, and so on, but there are a lot more there. And interestingly, they're gonna all contain this polar head. And when you look at the melting temperature, it always goes higher from the smaller number to higher number, especially when you make a comparison for those all saturated ones. And that is because this longer ones gets more stable, because it has a longer hydrocarbon tail, and those hydrocarbon tail can make the hydrophobic interactions between different fatty acid molecules. So it's going to stabilize its structure by itself. We'll have the higher melting temperature because it contains the lower internal energy. Question. So the question was that what actually makes this melting temperature changes based on the different number of carbons in your tail, right? Or the tail length. And I just briefly talked about the hydrophobic effect there. But can anyone answer to this question? Why? Because of double bonds. Double bonds. Oh yeah, yeah, absolutely true. So you're sort of answering in the indirect way, but it is a correct answer actually. So when we just simply focused on this saturated fatty acids, which do not contain any double bonds, you just think about this kind of long tail there. It's just a long tail. It's pretty stiff long tail. And it can be flexible based on the temperature though. But anyway, there's a long tail. There's another long tail of the fatty acid molecule. And they can actually interact with each other by that, like induce the dipole-dipole interaction, which is part of Van der Waals force. And it's a hydrophobic effect, right? And if you have a longer tail, then basically you have more place to interact with each other. So making themselves or each other more stable in that structure, which raises the stability of that state. So it's gonna get more like a solidified, like or the kind of raise the melting temperature because they're more stable. They need more energy to break apart those interactions. That makes sense? And double bonding is definitely important that we're going to talk about a little later. So now we're going to move on to the unsaturated fatty acids here, which contain double bonds. And here we saw the notation 16, colon 1, 18 colon 1, 18 colon 2, and so on. But here's a slightly different kind of notation here. Instead of using parentheses, you can also use n minus x here, and here there's an explanation here, and that's where the last double bonding happens within the hydrocarbon tail. So here 16(1) n minus 7 means that the last double bonding is seen at the carbon 7. Make sense? And of course it contains just one. But same thing applies to the this 18(2). A little fruit fly is bugging me here for some reason. But anyway, 18 dash colon 2, meaning that you're actually supposed to have two double bonds, which are at the C9 and C12. But you actually put n minus 6 here because the last double bonds that is seen is at carbon 12. And like I told you, you're looking at the last one actually counting from the last carbon. So that's going to be 18 minus 12, which is a 6, right? So n minus 6. The last carbon, the number of carbon. So you can actually put this total number of carbon. Like this one, you mean? So yes. So n minus 9, right? Because it's out of 18 carbons. So that's another type like notation that is not as useful as you actually list out. Yes. So you're counting from the last carbon, and going back to the bonding that you're seeing as a double bond, and you actually put that number there. So it's a little bit complicated. So I kind of don't like to use this notation. Linoleic acid. Yeah, that's a good question. So in case of linoleic acid, can anyone try? What would be the notation for this one, the n minus x? 18, 2, and then n minus? Can anyone? 6, because? Exactly, the last one is 12. So 18 minus 12, that's going to be 6, right? So again, this is a little confusing, right? So I don't like it, but this is another way to do that. It's not as informative as the using parentheses because you can't really simply see how many, where the double bonding happens actually. But it typically happens in cis. So you can, make a guess. You just subtract every 3 and then get those positions. But again, it's not a direct notation. But well, it is also used here. And when you look at the melting temperature overall is much, much lower than the saturated fatty acid, especially when you compare this stearic acid again to the alpha linoleic acid. This is minus 11, which is really, really low, right? And as you can see, as you have more and more of a double bonds, you're going to have even lower and lower melting temperature. And that totally makes sense because they're less stable in terms of their interactions with, the neighboring fatty acid molecule. So here we actually discuss a little bit more about the melting temperature or melting points of the fatty acids. But we already talked a lot about this. So I'm going to just quickly go through this. For saturated fatty acid, melting temperature increases with the molecular mass, basically the length of the hydrocarbon tail. And the single CC bonds are almost free to rotate and very flexible. And they can be easily packed together and making the pretty nice interaction between those carbons or hydrocarbons. Which makes this structure much more stable. Like all single bonds, extended conformation, more stable or the most stable because it has the minimal steric clashes between the molecules. And also they're going to have strong hydrophobic interactions to stabilize this structure, raising the melting temperature higher. So the shorter ones are going to be oils as opposed to longer ones are going to be solid fats again, because it depends on the total molecular mass or the length of the hydrocarbon tail. It's the same thing, right? For unsaturated fatty acid, melting temperature is lower than corresponding saturated fatty acid, again, with exactly the same number of carbons that comprises. And they do not pack well because the double bond makes a little kink. So that actually goes back to your. So relative, like comparing to the C18 to C20, then C18 is shorter, right? And of course, 14 is even shorter. Absolutely. So it's all about the melting points, right? Oils meaning that it's in a liquid form, and the solid fat is the solid form. So it depends on the where your the melting temperature is compared to the room temperature, right? Any other questions? Okay. This one? Mm-hmm. That's a really good question, actually, that I didn't even notice. But it can definitely happen. So the question was that compare this to EPA and DHA. DHA has more double bonding, so it's supposed to have the even lower melting temperature, which it should be lower than minus 54. So minus 70 or minus 80, something like that. But it's actually a little higher as minus 44. How come? Can anyone answer to this? Exactly. That's the great point here, where EPA has the 20 carbon in total, but the DHA has 22 carbons. So Now you are having sort of a balanced game where you have to compare the size of your tail as well as the number of double bondings, and that will affect in a slightly different ways, but in the opposite ways, right? So you just have to measure that. Until then, you can't really compare that. So it's not going to be a fair comparison. It also depends on the location of the double bonds too, where it makes that kink. And it can sometimes make the more dramatic kinks, in which case it'll create a bigger spaces, which inhibits the hydrophobic interactions between the tails. So that'll also, lowers the melting temperature. So that's all. That all depends. But as long as you're using the same number of carbons, now you can predict pretty well based on the number of double bonds. Again, this is pretty important where the double bonds introduce kinks. So when you have a double bond, it actually doesn't here. It's not here, but it will make the kink, the little angle there, which will create the space, bigger spaces. So it cannot be compactly packed as much as the single bonds or the saturated fatty acid does. Okay. And the soaps are basically the salts of the fatty acids. So here's the stearic acid, but the soap of the stearic acid would be just simply one losing the proton here. So it leaves the COO minus functional group here, carboxyl group here. And that's gonna be salt. And you use that quite a lot, right? It's even more polar. So it's gonna have the amphipathic kind of property where you can, use it's dissolved well in water, but also, attached to the fats or the lipids pretty well. So you can, clean your hands basically pretty easily with water. And fatty acids, these are rarely found as a free form in nature, but it's usually esterified. Esterified meaning that I'm using this carboxylate group to attach to the other molecule, which is these triacylglycerols, glycerophospholipids, or the sphingolipids. These basically use these fatty acids for their tails, but as you can see, these molecules contain multiple tails, and each tails are basically fatty acids or some other functional groups. So triacylglycerols are based on the glycerol, but it has 3 fatty acids there and can be used as a storage form. And the glycerophospholipids have a similar structure here, glycerol with the 2 fatty acids as tails. But instead of have another fatty acid, it contains the phosphoryl group that is attached to alcohol, basically. And alcohol can be pretty long, short, and so on, as long as it contains hydroxyl group, right, OH group, so it can lose the proton. And sphingolipids have a particular head group here too, instead of just the glycerol. It has 1 fatty acid and some other functional groups that is linked by oxygen here. And especially glycerophospholipids and the sphingolipids are important because they are the critical components of the lipid bilayer, the biological membranes, as opposed to triacylglycerol isn't. It's only the storage form. And that is, again, really important. We're going to go over these different types of lipids now that has the multiple fatty acids as their tails. The first one is triacylglycerols, or TAGs, and there are triglycerides of neutral fats. So here's a glycerol. It contains simply 3 carbons, and all are attached to the hydroxyl groups. And instead of having hydroxyl groups, it can have this COO region, which is the carboxylate of the fatty acids, right? So it can be esterified to that to contain R1, R2, R3, basically 3 different types of fatty acids there. It doesn't have to be the same, R1, R2, R3, but all different kinds of things. So here's an example where you have 1 palmitoyl oil, 2 linoleum oil, and 3 stearoyl oil glycerol. So basically you put oil OYL, it kind of is a tongue twister for me. But anyway, put the different kinds of fatty acids there to form this triacylglycerols. And here are some more, properties of the fat, especially those kind of fats that have sort of a more complex structure than just the fatty acids. And we're going to make a simple comparison with the carbohydrates here. The fats in general, especially for those, glycerophospholipids or triacylglycerols and things like that, function as efficient energy reservoir in animals. And it yields more energy upon breakdown than sugars do. And sugars, I'm talking about the carbohydrates. And the comparison here is the 9 versus 4 kilocalories per gram. So it's at least twice much or more, right? And usually these triacylglycerols, especially in the storage form, make the anhydrous form, which means that it actually excludes all waters there. And that means it excludes water, which is pretty a lot everywhere in the cell. So it's going to be more compact because none of the other molecules, especially water molecules, involved in this, making this a molecule, as opposed to carbohydrates involves a lot of water molecules. It gets into that, works as buffers and so on, or stabilize the structures and so on. And the sugar itself is pretty well dissolved in water, so in most cases contain water. So it can be compacted as the polysaccharides, but still will contain a lot of water molecules in it. So it's not as compact as the fat. And more compact meaning that it requires less room to store the same amount of energy. And that's why it is more efficient in the storing energy. Fats provide 6 times more metabolic energy than equal weight of glycogen. It's the fact, but based on this feature here. And fat reserves allowed to survive starvation for up to 3 months. So it's going to be slowly metabolized, but can create, provides a lot of energy there. And adipocytes, or the fat cells, are the one that generates the fat or the stored fats that we already talked about. So it's specialized for synthesis and storage of especially triacylglycerols. Again, triacylglycerols are the storage form of the lipid and serve as insulation layer in whales and Arctic animals, and for humans too, on your belly and stuff like that. And the triacylglycerols are most abundant class of lipids, but are not, again, I'm saying this multiple times, not found in biological membranes. It just serves as a storage. The next class that we talked about is the glycerophospholipids, also called phosphoacylglycerols, or glycerophosphatides, or the phosphoglycerides, and so on. But in most cases, we're going to just call them glycerophospholipids in here. And that's a subclass of phospholipids, but obviously contains glycerol, base there. And it's a major component of the biomembranes. And the fatty acids that are attached to them, the esterified, happens at the C1 and C2 position, because at C3 position, it always contains phosphoryl group, and that's why it is called phospholipids or glycerophospholipids. And as you can see, there's a glycerol here. That's why it's called the glycerophospholipids. The simplest structure would be the phosphatidic acid, which I don't have structure here, but basically that structure is same as this one, glycerophospholipid. And instead of having X here, it has hydrogen. That's all. So it's a pretty simple structure. And you can have all different kinds of functional groups on here, replacing this X position here to create the like divergence in the glycerophospholipids. Question? Name of the what? Which one? Oh, that is the phosphatidic acid. So that's the simplest form I'm talking right here. Sorry, sometimes I just talk really fast or jumping back and forth and things like that. So I'm actually on this right here, bullet point. Simplest form is the phosphatidic acid, which is X, the hydrogen for X. Yes. What does what? Oh, esterified or ester bond is basically C, O double bond, and O here, bridge. That's the ester. And esterified meaning that it has a bridge or the bonding or connection using this functional group, ester bond, ester, functional group. Any other questions? Okay. And as you can see here, the phosphoryl group is not esterified, meaning that it does not contain COO, right? But it only has just O here, which is negatively charged. So the X can be just hydrogen to form the phosphatidic acid, or we're actually talking about this phosphatidic acid specifically, or it loses this hydrogen, which will become negative, negatively charged. So it can work as a sort of an ion. So it can interact with the cations like sodium, potassium, calcium, magnesium, and so on. But it's just a minor amount is observed in the biomembranes. I'm not talking about the overall glycerophospholipids. I'm just talking about phosphatidic acid, the simplest form. And phosphoryl group is usually esterified now to the many different kinds of functional groups to make different kinds of GPLs or the glycerophospholipids. And these are some examples here. When it's attached to choline, then it's going to be called as phosphatidylcholine, where you can see the same structure here, right? Glycerol-based structure here. Two fatty acid that are attached, working as the tails. And what is choline? Choline is this: CH2, CH2, and CH3 triple. What kind of molecule is it? Well, I don't know. I mean, how to answer to that question. What kind of molecule the choline is? Choline is the molecule that is just widely used as a functional group, especially for the lipids. They can be derived from, as it contains the amino group here, not the actual amino, but the nitrogen here. It can be based on the amino acids, so it can be produced in that way. But mostly, it's just abundant there, where you can just attach it, conjugate to to generate these phospholipids. Was that an answer to your question? It's just but its structure is right here. And also, it can be phosphatidylethanolamine. So here's ethanol, here's amine group, right? So it's slightly different from this choline, where it has 3 hydrogen atoms instead of 3 methyl groups. Again, pretty abundant in the cellular system. And also, you can see the all different kinds of like a phosphatidylserine. Serine is attached there, and serine, as you remember, its R group is a CH2OH, and this O is used to esterified or the link to this phosphate. And this is again called esterified because it has POO structure, right? As long as it has some like atom and then OO, especially one O with the double bonds, it'll be called esterified, and so on. So, and inositol can be used. So based on the what kind of things, functional groups attached to those phosphoryl group of the glycerophospholipids, you can actually generate all different kinds of classes, and they're going to be involved in slightly different manners in the biochemical reactions, but basically forms the membrane. We kept talking about the glycerophospholipids. And here, we already talked about phosphatidylcholine and phosphatidylethanolamine, which has a similar structure. Therefore, their X group are two most common constituents of the biomembranes. And they are all amphiphilic or amphipathic because they're, the group, the phosphoryl group is charged or have a polar, properties. So it's going to have a polar head, and the two fatty acids will serve as nonpolar tails. And it's spontaneously formed micelles or lipid bilayers, and we talked about these, two different things, and we're going to talk a little bit more later this lecture too. And these are the basically the choline, serine, or the ethanolamine structures here, and how they are called when they're attached to the phospholipids. And the phospholipase is an enzyme to cleave these phospholipids. And here's an example of this lipase or the enzyme working to make detach the one fatty acid. So phospholipase A2 specifically attacks the R2 group here. And then with the involvement of water molecule, so it's a hydrolysis basically, and the detach this functional group or the one fatty acid. And also, like there are different phospholipases, like phospholipases A1 detaches the R1 group, and the C gets cleaved on this region of the phosphoryl group, and the D also cleaves the phosphoryl group, but in the different place basically to create the lysophospholipid. Okay, any questions so far? Again, these are just a lot of facts, but they're going to be pretty important in the end to understand totally about the how lipids are metabolized or used for forming the biochemical, like involved in biochemical reactions and forming all the important organelles, especially the plasma membrane and the other organelle membranes. The next one is sphingolipids. Again, this one has multiple tails, but the use the sphingosine or the sphingosine derivative as its head group. So it's also a major component of the biomembranes, like I told you, and it's basically derived from the sphingosine, where you can see the structure here. This is simply the C is at C18, 12, 13, 14, 15, 16, 17, 18. So C18 structure, oh, it actually says here, 18C amphipathic, because here's this seemingly looking like a tail is hydrophobic, but here's a hydroxyl group, here's another hydroxyl group, and the amino group makes this other region charged or the polar. So it's an amphipathic group, especially for this amino alcohol group. And there are many derivatives, and there are basically ceramides where the other fatty acids are attached to this amino group. So ceramides derivatives are again basically sphingolipids, and these three are sort of major components again form the biomembranes, which are sphingomyelin, cerebroside, and ganglioside. And as you can kind of guess, based on their names, myelin, cerebellum, ganglions, and so on. Those are all kind of neurons, right? Nerve cells, nerve cell types, and so on. So these lipids are most widely observed in the nerve system. And also, of course, sphingomyelin will be observed in myelins. Gangliosides will be in ganglions and so on. So their head groups are different, basically. So for the sphingomyelin, It can use phosphocholine or phosphorylcholine, phosphatidylcholine. We already talked about this, right? Or the phosphoethanolamine. So hopefully you're a little familiar with its structure. And for cerebral side, it uses just a single simple sugar residue like glucose or galactose. And for the ganglioside, which you can see the structure right here, has a little bit more extended head group. It has the oligosaccharides with at least one sialic acid. The sialic acid here is attached to the second sugar here. And based on the how many sugars you have in your oligosaccharides, you can actually categorize these gangliosides in a different ways. And here's a structure of sialic acid again, based on this six-membered ring, which forms the sugar. And there are lots of carboxylate or the hydroxyl groups attached to that. So it'll create a lot of will give a lot of clarity there. So the first one we talked about is a sphingomyelin. And that is basically ceramide that we talked about, that structure have two fatty acids, as a tail, and then have the phospho compound attached to the end of hydroxyl group, which is right here. Again, it has two tails, and one tail contains a phosphoryl group instead of just a hydroxyl. And it has this choline again structure here. And it can have the phosphoroethanolamine again. Instead of having N and triple CH3, it can have just simply NH3. And that's a component of a myelin sheath of nerve cell axons. And axons, as you know, is wrapping around the processes of the nerve cells to quickly transfer or transduce the electrical signal there. And it insulates electrical impulses, so that's definitely important for axonal excitation. And myelin sheath breakdown occurs to the multiple sclerosis. So it has a relevance to the human disease, of course. The second one is glycosphingolipids. For some reason, it's working or not. I don't know. Just to make sure that it's well connected. Okay. And we quickly talked about the cerebral side and the ganglioside here. And the important feature of that is that it uses a sugar, a simple sugar, as a head group. So it is replaced, for this hydroxyl group of the ceramide. And cerebral side, gangliosides, as well as the sulfatides are the components of the membranes, especially in nerve cells. And cerebral side are the ceramides, again, derivative with the simple sugar head group like glucose and galactose. Gangliosides, again, we talked about, has the oligosaccharides as the head group, but contains the sialic acid, which have the negative charges. So I'm talking about this twice, right? And this sugar group of the cerebral side can be extended outside of cell surface. And that actually is a really important feature because that serves a lot of important roles. It can work as a receptor for the pituitary hormones, or can be work as a determinant for the cell recognition because it has that specific features there, the sugar group that is attached there that can be recognized by the other proteins. And also work as a receptor for the bacterial toxins and so on. An example is cholera. And when you look at the ganglioside, I told you that there could be different number of sugar molecules can be used for forming that head group. And here, they are called GM1, GM2, GM3, and so on. And this G refers to gangliosides, and M refers to the monosialic acid. So here's one sialic acid that is attached, so it's going to be mono. So M is for mono. And 1, 2, 3, these numbers are based on the relative mobility in electrophoresis for the, as you know, when you do the SDS page gel and, you can have the different mobility based on their size, right? So it's basically M1 being the biggest one because it moves the least, as opposed to M3 would be the one that moves the farthest, and that's the smallest one. I know it's counterintuitive. So GM1 is basically refers mono, sialo, tetrahexosyl because there are 4 hexos there, ganglioside. And it can also contain the 2 sialic acids attached to other molecules, other sugars, in which case you're going to call it GD and 1, 2, 3 based on what you have. So that's going to be di-sialo, and it can be tetra or tri or di based on the what number of sugars you have and ganglioside. Okay. And the other classification, we're going back to the sort of the like first, second, or third slide, where we talked about all the different classification of the lipids. The waxes are also widely used in living organism. That's an ester. Again, ester is the COO of the alcohol and fatty acid. And that is actually a waxy coating substance for the fruits like lanolin. And terpenes are also widely used. It uses the isoprene as its unit, base unit, which structure is written here, the isoprenes here. And there are many different kinds of derivatives of the terpenes, as you can see here, limonene, and the squalene or the lycopene and so on. And they have all kind of intense flavor, like distinctive color, odor, and things like that. Examples are menthols, citronella, and things like that. And the steroid, I believe, is the last class of the lipids that I introduced at the beginning. And steroids have this sort of a tetracyclic structure where the 4 carbon rings are fused together to form this peculiar structure. And cholesterol, which is right here, is a component of animal membranes and precursors of steroid hormones. And it serves as a derivative or the precursor of the many steroid-based hormones like testosterone, estradiol, or the progesterone or so. And cholesterol is really important because it's a component of lipoprotein complexes, and that serves as the transporting the lipids in the body from the energy source to the region that has to be consumed, or the vice versa, the other way around too. And there are different types of lipoprotein, and you probably are familiar with at least a few of these. First one's chylomicrons, which is the least dense lipoprotein complex, and that is responsible for transporting the dietary triacylglycerols. Again, this is just for storage, right? And cholesterol from intestine to liver. So as soon as you consume or intake the cholesterol or the triacylglycerols or other lipids, they're going to be trapped in chylomicrons and then will be transferred to liver. And this VLDL, or very low density lipoprotein, is responsible for transporting triacylglycerol from liver to tissues. And LDLs and HDLs, I'm pretty sure you're familiar with it, low density or high density lipoproteins. The LDL is considered bad cholesterol. It contains about half of the cholesterol there, 50% consists of cholesterol, as opposed to 25% would be protein. And that is transporting the lipids from liver to tissues. And that is bad because you're having a deposition of the cholesterol to the extent. So having too much of a cholesterol will cause problem. that again we're going to talk about. As opposed to HDL, the high density lipoprotein contains much lower cholesterol, but larger amount of a protein. And it also transports these proteins or the lipids from tissues to liver. So it's a good cholesterol because it's scavenging. It collects the cholesterol from the tissues and basically send them to a liver for the disposal. So you're getting rid of excess cholesterol using HDL. And LDLs are more susceptible to oxidative damage, and damaged LDL deposited in arteries to cause the disease, atherosclerosis, basically, artery hardening. And this is a structure of lipoprotein where it contains the different kinds of lipids containing triglycerides or the triacylglycerols, as well as other lipids like phospholipids and so on, and lots of proteins. Again, the lipids, the most critical function, one of the most critical functions of lipids in the living system is forming the biological membrane. And that basically defines the inside and outside of the cell. And like we discussed, it is important for compartmentalization of even within the cell for the specific biochemical reactions, and also serves as a venue or the surface area, surface region where all other reactions can occur there too, because there are a bunch of other proteins or the lipids or some other sugars and so on are all kind of placed there. So it's a nice place where all those different molecules meet each other and do some kinds of reactions. And an example of that is the electron transfer chain, or ETC, in mitochondria that works for the respiration, generating the energy there. And the membrane is assembles of the lipids, proteins, and carbohydrates, or pretty much everything. And there are different classes of proteins that are involved in forming the membrane, like integral proteins, channels, or the transport, or also the peripheral proteins and so on. And some proteins are decorated by the sugars, as we learned last lecture, like glycoproteins or the glycolipids and so on. And again, glycerol, phospholipids, and sphingolipids are important because those are critical components of plasma membrane or the biological membrane. And again, as we talked, this is amphipathic because of those their head groups that are polar. And we already talked about this. It makes the micelles or the bilayers spontaneously. And it depends on the conditions, the environment that those molecules are under, or also the temperature and the molecules that are involved for forming these micelles or bilayers. So for example, if you have just a layer of water and the another layer is basically air, it's just exposed to the kind of free space, then these lipids will form, basically coat all the water molecules, create the really nice monolayer structure there, regardless of their constituents or their shapes or the structures and so on. As opposed to if there's a little water droplets there, then all the polar groups will face the water molecule, as opposed to the tails, which are non-polar groups, will try to be, stay away from the water, creating this kind of micelle structure. And same thing applies for the other way around here, where there's a water molecule surrounding those lipids. Then it'll try to expose these polar groups outside. So again, facing the water molecules, but hiding their non-polar regions, and put it away from the water molecule. So we'll also create this micelle, but inverted micelle basically. And you're going to see mostly see the micelle in this structure in the living system, because it's just all waters, right? And same thing applies here, where the lipids are surrounded by water everywhere. But instead of forming the micelle, if it has a sort of a bulky tail, bulky hydrocarbon tails, then it's hard to form it as this kind of nice sphere. So instead of forming a sphere like this, it will just align nicely there to form some kind of layer there. And of course, you have to make the bilayer to hide these nonpolar regions from the water molecule or water aqueous environment. So it'll naturally create this bilayer. And all these processes again happens spontaneously. Any questions so far? Okay, we talked about the micelles and lipid bilayers already. Here, quickly, micelles is the structure defined by the critical micelle concentration and the aggregation number n. And these are basically determined by the type of lipid molecules that you're using for forming this micelle structure. And general rule is that if you have a longer tail, you're going to have a higher aggregation number. And that totally makes sense, right? Because longer tails, meaning that you're having higher hydrophobic interactions. Just a fly just bugging me constantly today. And you also have to have a single tail because the space matters here, right? To form this spherical structure. If the tails are bulky, like having bitails or triple tails, not really, that's a possibility, bitails, then just like the glycerophospholipids or the sphingolipids, then that's gonna form the flat bilayer structure like this. And also, these lipids can form liposomes. And liposomes looking similar to micelle, which creates a huge spherical structure. But instead of having the all hydrophobic regions hidden inside, it actually creates a little like cavity, or not the cavity, but the sort of a vesicle. So they're going to be a aqueous space inside of this structure, which can contain some like polar substances, just including water, as well as some soluble proteins and so on. And those vesicles are called liposomes. And they are stable structure, so they can be purified by gel filtration. And basically, they can create the inside and outside compartmentalization. So, and they can be different because this inside is completely trapped and surrounded by this lipid bilayer. And you can basically generate these liposomes and then insert some proteins in interest that are interacting with lipids, such as channel proteins and so on, to study those like behavior of those proteins, because it's like mimicking a small like real kind of cell condition, right? So you can actually test that, what it induces, that transportation, and how it behaves depending on what kind of mutations introduced and stuff like that. And also, you can use this liposome as a vesicle for your drugs or the gene therapy. All right, so here's the membrane structure. It is actually dynamic, so it's fluid-like, and you can imagine that there are two types of diffusions happening. One's lateral diffusion, where it's just along the same one single layer of that bilayer structure, and it goes just alongside the lateral sides here and there. As opposed to transverse diffusion can happen too, but it's really rare, where the one lipid molecule flips over to move to the other layer of that bilayer structure. So it requires a lot of energy and takes basically really doesn't happen that much. So there are enzymes that are involved for such diffusion, the transverse diffusion, flipases or the translocases. Flipase is passive, meaning that this enzyme only just helps the flipping the lipid molecule, but it's not involved in like inputting some energies to accelerate the process. It just happens there around this protein, creates some favorable condition environment for flipping the lipid molecule, as you can see here. So it just flips and goes like that. As opposed to translocases are the consuming the ATP, basically use energy to actively flipping over the lipids. And that is especially important for controlling the constituents or the ratio of the different kinds of lipids in the membrane. And that confers a lot, on those lipid bilayers, especially when you're talking about different kinds of organelles and so on. You have to use the different composition of the lipids to precisely regulate their functions, stabilize the proteins that are integrated, and so on. And this membrane, again, works like the sort of a fluid. So since it is just simply oils, right, the lipids, it can experience phase transition. So it's normally gel-like a structure, but when it is heated, then now it can get loose because, overheated. And so now they have more dynamics, especially for those fatty acid tails. So they can flap and, fling a little bit more, making more like a liquid kind of behavior there. So here's a before transition where it has a more like a solid structure. When they are stacked together really nicely, so it's more like a solid form, right? And there are lots of hydrophobic interactions occurring to stabilize this structure, as opposed to after heating, then these tails can flap here and there, making these different conformations to create more space, basically inhibiting some of the really kind of nice, stable hydrophobic interactions, making it more like a liquid. And this fluid-like a state, or the phase transition of a biological membrane, is a function of composition, the type of lipids that are involved for forming this membrane, and also temperature too. And again, phase transition is about the organization, involve the changes in organization of fatty acid tails, not the head groups, right? It's just the tails and their conformations. And it's a fairly sharp transition where it happens around this melting temperature. And it totally makes sense, right? Oil form versus solid form. And below the melting temperature, they're going to be tightly packed. So it's gel-like, and there's a little motion. As opposed to above the melting temperature, it's going to be behave like a fluid-like. So because the tails are more mobile, and also creates the more space there. And this melting temperature is influenced by the chain length that we talked about. The longer the chain, the higher the melting temperature. And also degree of saturation too. So we talked about this, that the double bonds creates the kink, right? Again, making more structure. It's a rigid structure, making more space in there, inhibiting some nice hydrophobic interactions there, which will, lower the melting temperature. And also cholesterol content is important for making the different states, the phase transition of a biological membrane, because cholesterol has the rigid ring structure. So that means it's rigid. So if they are embedded in the membrane, it will decrease the fluidity or increase the rigidity. So more cholesterol means that there's a high melting temperature for that membrane structure. And also, very high contents of the cholesterol will abolish the phase transition completely, because the tails cannot, get close, because there's a rigid, like wedges, like cholesterol wedges are everywhere. So there's a little chance that these more flexible fatty acid tails can, meet each other to form the nice like interaction there, hydrophobic interactions there. But there's an interesting relationship between the temperature and fluidity of the cholesterol itself. So this is one graph here. So without cholesterol, you're going to have higher fluidity as the temperature goes up, and it changes dramatically around this melting temperature. But if you add cholesterol to your membrane, now at the low temperature, as you can see, it raises the fluidity. Because instead of just a complete solid form, the cholesterol itself is pretty rigid, but also inhibits the interaction between the fatty acid tails. So it'll actually relieve a little bit of interactions there, or relieve the energy there, inhibits the interaction, make the fluidity goes up a little bit. Um, as opposed to at high temperature, it decreases the fluidity because now you have lots of rigidity there built up. So it is supposed to be like really nice liquid, but lots of, stiff crystals are embedded there. So it can drop down the liquid fluidity. There's a asymmetric structure of the membranes, bilayer membranes, when you look at the membrane structure. So this is a bilayer structure. This is the inner membrane. This is not the inner portion of bilayer. This is the outer portion of bilayer. And they have a different composition there. And that composition is pretty well conserved, actually, have a different, there. And the flip pages or basically the proteins that are involved for regulating composition is important to keep this asymmetric structure of the lipid bilayer. And that is also important for creating the different charges inside and outside of the cells, or some other regulations, and there are proteins working inside the membrane as an integral membrane. So this is the last slide. And the Singer and Nicholson proposed this fluid mosaic model to explain how the membrane, biomembrane behaves, which was proposed in 1972, and it's widely accepted these days too. There are different classes of proteins are involved in this model. Intrinsic or integral proteins are the transmembrane protein basically embedded inside the lipid bilayer. And they interact strongly with the bilayer, so they're going to have hydrophobic regions in the middle, right? To be stably just embedded there. And there are intrinsic or peripheral proteins here that will interact more like the head group or the polar group of the lipid bilayer. So it's more loosely associated and can be easily removed. And the interaction is basically based on electrostatic or hydrogen bonding again. So instead of polar regions that are playing a critical role for the integral protein, peripheral proteins uses the polar regions to create this kind of interaction with the lipids, lipid bilayers. And proteins embedded here, integral proteins or peripheral proteins, are pretty much dynamic. So they constantly move by the diffusion along this lipid bilayer. And they move really fast, like a few microns per minute. And imagine the size of the cell. The size of cell can be ranges from like a few micron actually to hundreds of microns for the bigger like mammalian cells, but can takes the only minutes to across all those membranes along the within the even the big cell. And phospholipid molecules move from one end to the end in bacterial cells less than 1 second because bacteria is really small. All right, so that's it. And again, important thing is the lipids, types of lipids, how they're involved in forming the other like important organelles in the cell, like membranes.